And at that elevation in that part of Mexico, there are these gorgeous forests made up largely of fir trees. And the scientific name of the fir tree is Abies religiosa. And it's called religiosa because the feeling that you get when you're in these groves of the fir trees is very much like being in a cathedral. So the monarchs start roosting on the branches of the fir trees.
My guest today is Dr. Doug Taron, Chief Curator Emeritus at the Peggy Notebaert Nature Museum. Thank you for joining me, Doug.
Oh, good to be here. Good afternoon.
So how did you get started in the field of biology?
Well, it probably was through butterflies because when I was 6 years old, I got a butterfly net from the Easter Bunny. And that's something that I've carried with me all of my life. And so it's just, been an interest since very early childhood.
It's just continued on.
With some detours, but...
Yeah, and specifically butterflies because of the butterfly net.
Well, that's what sort of was my my entry point into all of this.
Could you share with us the trajectory of your education?
Sure. I majored in biology as an undergraduate. I went to a little liberal arts school, Colby College in Maine. And then when I went to go to graduate school, I decided to go into biochemistry. And went to Northwestern, pursued a doctorate in biochemistry. When I was there, I was studying microbiology problem. I was looking at the way certain types of bacteria synthesize a particular kind of macromolecule that sits on the surface of the cells. So that took me into more of a biochem, biotech, molecular biology direction. And after grad school, I worked for about a dozen years in the biotech industry. I was at that time researching labeling technologies that allow use of DNA as a diagnostic tool for things like cancer and infectious disease. While I was doing that, I was still interested in butterflies. And I also began in the early 1980s working as a volunteer doing ecological restoration at a nature preserve here in Elgin. And in the late 1980s, that work was being overseen by the Nature Conservancy at that time. And in the late 1980s, the staff at the Nature Conservancy were realizing that most of this ecological restoration work involved plants. People were doing things like removing invasive species, gathering and scattering seed of native species, reintroducing things like fire onto the landscape. And all of those are primarily directed at the plants, and they were wondering what would happen with the animals. And at that time, a gentleman named Ron Panzer, faculty at Northeastern Illinois University, was who they approached to figure out how to answer that question. And they wanted to look at what was happening with animals on nature preserves in a way that it could be done by people who did not necessarily have formal science training, and that would be a long-term project that could produce quantitative data. And Ron looked into some work that was coming out of England that was developing techniques for monitoring butterflies. And that was landed on as a very good group to work with. And of course, because of my long-standing interest in butterflies and because I was very active on one of the nature preserves near here, I wanted in right away. And so I worked for about 2 years just collecting data as a volunteer collecting data. About 2 years into the program, Ron said, I don't really have the bandwidth to do this anymore. This Taron guy seems to know what he's doing. Why don't you see if he will volunteer to run the program for a while? And so I thought that sounded interesting and agreed to do that. And for about 10 years, I ran the Illinois Butterfly Monitoring Network as a volunteer.
While maintaining your job in biotech?
While maintaining my job in biotech, yes. The program was much smaller than it is today. I don't think today I would have been able to do it with the size of the program as it is today. So in the 1990s, the Chicago Academy of Sciences, which is the parent organization of the Peggy Notebaert Nature Museum, was planning on building the new museum. And I knew the then president, Dr. Paul Heltney, and he approached me and said, "We're building this exhibit about butterflies. Would you be interested in taking a look and commenting on the plans for me?" And at that time, I was sort of ready for a career change. And I was looking at the plans and thinking, if you put a research component into that, that could be kind of a sweet deal. And so I called him the next day and I said, "Well, you asked me to comment on the plans, and my first comment is, are you hiring?" And he said, "Are you interested?" And I said, "Well, maybe." So he said, "Okay, why don't you come in? We'll have lunch together and talk about it." So I kind of laid out what I thought an interesting research program that could go along with the butterfly exhibit might be. And a few months later, they hired me. So that was sort of the path that I took to getting into the museum.
Nice. Can you share a little bit about your research on butterflies, and specifically what aspects of butterfly research are you looking at?
There have been two main components to my butterfly research. One of them has involved, it was sort of an outgrowth of the work that I was doing on ecological restoration. And a lot of these preserves are now so isolated because Illinois is so badly fragmented that even when you make the plant communities better, there's no place sufficiently close by for butterflies to repopulate the site from. So you have to bring them back deliberately. And that was one of the things I was very interested in doing at the museum, setting up a laboratory where you would go out to remnant populations of some of these rare butterfly species, bring some into the laboratory, rear them, and then release them on a new site that's one of the ecological restoration sites that's under management. So a lot of the work that I've done has been here in Elgin at Bluff Spring Fen, which is the site that I have been steward at for, since 1982, so a long time. And the way this works is you collect on a donor site a modest number of female butterflies. So you first have to make sure that the donor site has a sufficiently robust population that you can take a half dozen females without damaging the donor population. And pretty much any time you catch a female butterfly in the wild outside, she is already going to be fertile. She's already going to be able to lay fertile eggs. And depending on the species, there are different types of cages that we set them up in, but the general setup is that there is a small container that has sprigs of the host plant. It has a means of introducing like Q-tips moistened with diluted honey for the butterfly to eat. And the butterfly will then lay eggs in the cage. And how many eggs you get out varies with the species. So one of the rarest species that I've worked with, the swamp meadowlark, you're lucky from 5 or 6 butterflies if you can get a total of 200 eggs. Another species that I've worked with, the regal fritillary, has a much greater fecundity than that. And it's not at all unusual to get 700, 800, 900 eggs out of a single female. So you collect the eggs out of the cage, and we generally put them in petri dishes on moistened filter paper and let them hatch. And then you depending on the species, you're going to start feeding the caterpillars. Now, one of the species that we've worked with, the regal fritillary, is a little challenging because these butterflies are big. They're about the size of a monarch. And yet the eggs are tiny, tiny, tiny. And for that species, the eggs hatch in September. And then the caterpillar, immediately goes into hibernation. And so you're challenged with keeping these tiny, tiny caterpillars over the winter. And then in the spring, when they emerge from hibernation, you start feeding them. Most of the species we've worked with, they hatch during the summer and you feed them for a while and then they go into their hibernation. And then you complete feeding them the next spring. They pupate the adults emerge, and then we transport them out to the release site and let them go. And with luck, they will start a new population.
And how do you differentiate a male from a female butterfly?
Some species, the wings are very different colored, or at least different enough that you can tell them apart easily. Some species, you actually have to catch them in the net, take a close look at the tip of the abdomen, and see if they have an ovipositor, which means it's a female, or claspers that mean it's a male. And we just keep the females.
Why are butterflies such important indicators of environmental health?
Well, just about any group of organisms is going to be an indication of biological health. Insects are a particularly good one to work with because they're very species diverse. So on a nature preserve of several hundred acres, which would be the size that would be, by Illinois standards, that's a fairly large nature preserve, you might have a dozen species of mammals, but you will have hundreds and hundreds of species of insects. And so, you can see a lot more about the biodiversity on a site by looking at insects. Butterflies are good to work with because they're really easy to study. They're big. You don't need specialized equipment to study them. A lot of things are really tiny, and you have to look at them under a dissecting microscope, and they may, as a group, not be terribly well studied and be very difficult to identify. Butterflies have none of those problems. There are tons of field guides. They're active during the daytime. So, there are a lot of features of butterflies that make them a really good study group for these types of questions. So what you're going to find is that, first of all, a parcel of land that is in good shape from an ecological perspective is going to tend to have more species on it. And more importantly than that, the number of species on the site is not going to decline over time. And unfortunately, not only we here in Illinois, but people around the globe have been finding that when you study these things, we are losing both species diversity and abundance of butterflies on preserves all over the planet. And so that is a little bit distressing.
Yeah. Let's talk about the monarch butterfly, because I think that's obviously one of the species that I think a lot of people are familiar with. And could you talk a little bit about their migration from northern United States and North America down to Mexico?
Yeah, the monarch is absolutely fascinating butterfly, and the migration is a big part of that. And east of the Rocky Mountains, you will have monarchs up here during the summer months, and they will have several generations each year. And starting in early August, the generation that emerges from the chrysalis around that time of year starts behaving very differently from the previous generations that summer. And they're a little bit bigger. They start aggregating. Monarchs for most of the summer are very sort of solitary, and other than mating, they don't really interact with other monarchs very much. As you start getting into late summer, the monarchs start roosting communally at night, and sometimes you can go to particular places and you'll see that there are monarchs all over the bushes or the trees. This is the beginning of the migration. They'll begin flying preferentially in a south to southwest direction, and they're going to Mexico. And the wave of the migration, so they'll start earlier in the year further north into Canada. They range into the maritime provinces in Canada. So earlier in the year, or further north, they start the migration behavior a little bit earlier in the year. The peak wave of migration crosses the Chicago area generally somewhere in the first half of September, and then tapers off. You will see monarchs, but in lower numbers, into October, or even a few of them into early November some years. And the further south they go, kind of the more concentrated they're getting into like Texas, and they continue flying until they get to the mountains about 100 miles west of Mexico City. This mountain range is called the Transvolcanic Belt. transvolcanic because the range is situated more east-west, and the major cordilleras in Mexico are north-south. And they go to high elevation. They go up to about 10,000 feet. And at that elevation in that part of Mexico, there are these gorgeous forests made up largely of fir trees. And the scientific name of the fir tree is Abies religiosa. And it's called religiosa because the feeling that you get when you're in these groves of the fir trees is very much like being in a cathedral. And so the monarchs start roosting on the branches of the fir trees. And there were so many of them at that point. You've taken all of the monarchs in the eastern North America, the US and Canada, and squished them down into about 20 sites, each of which is maybe a few dozen acres. So, you know, they're really all over each other, and they just, they weight the branches down because there are so many of them. And they'll land on the tree trunks, and you'll see a tree trunk that's just monarch, 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 and no space where you can see the bark of the trees. And they will stay in the mountains of Mexico for the entire winter. Now, these monarchs that are in Mexico are the ones that emerged from their chrysalises up here in August. So they come out in August, they fly down to Mexico, they get there around the 1st of November. They stay the entire winter in the mountains of Mexico. And then sometime in March, they start becoming a little bit more active. There's one other thing about these monarchs, unlike the earlier generations, is that they're not exhibiting reproductive behavior during the migration and during most of the overwintering. And in March, they emerge from that and they begin mating, still in the mountains of Mexico. And somewhere around mid-March, they start leaving and they start flying further north. And they get to about the Rio Grande Valley. And that is really the first place north of their wintering grounds that they're going to encounter the milkweeds that the caterpillars will feed on in any of abundance. And there's a mass egg laying in the Rio Grande Valley at that time of the year. And then most of them, finally, many months later, after they emerge from their chrysalises, will die. And it is the first or sometimes even the second generation on the way back that are the ones that make it back here. They get back here right around Memorial Day each year.
And how do they navigate? How do they find their way?
Boy, that is one of the big mysteries of nature. And it's also one of the things that keeps studying this as interesting as it is, because there's still so much that we don't know. So we know a little bit about it. We know that it works in part with their circadian rhythms, their biological clock. And we know that they use the position of the sun in the sky to navigate. So there's a solar compass involved in this. So their biological clock tells them, you know, roughly what part of the day they're in, how far from noon they are. And they will orient towards where the sun is at its zenith during the day and start flying more or less in that direction. That doesn't tell you a lot about how they navigate, because they get to the same places every year. And it's sort of like if I were to say to you, there's a particular parking lot in Zitácuaro, Mexico that I would like you to go to. Here are the keys to my car. South is that way. Good luck. And that's all the information that you have. That is sort of where our understanding sits at the moment. Now, they use natural contours. They will often use rivers and coastlines as navigational corridors. And one of the things that happens with coastlines is that sometimes the coastline serves, the details of it serve to concentrate a lot of the monarchs, and you'll get roosting sites where they kind of wait for favorable winds to cross a body of water. So Point Pelee on the northern shore of Lake Erie is a good example of that. They will kind of pile up there in large numbers until they get favorable winds and then cross the lake. Lake May, Cape May, New Jersey is another very well-known place where that happens, and you know they're crossing the Delaware Bay there.
How many days does it take for them to get, let's say, from the Chicago region down to Mexico?
So they will, sort of the peak of the migration here is in the first half of September, and they really start getting to Mexico in early November. So it's about 2 months from here to Mexico.
And do we have any idea of how far they fly during the course of a day, or?
This is something we're just starting to find more about. So the way we knew that it was the same for a long time, that it was the same butterflies making the whole trip, is through tagging. And the way tagging works is the tags are little bits of paper with adhesive on one side and data on the other. They're about the size of a paper punch. And they're affixed to the middle of the hind wing of the monarch. And they stay on, and it doesn't seem to interfere with the butterflies flying. And they're released, and off they go. Well, monarchs that have been tagged in Canada have been recovered in Mexico. So the only way that that's going to have happened is if the same butterfly was able to make the entire trip. Mm-hmm. We're starting to be able to do stuff with actual radio tracking. Radio transmitters have gotten so miniaturized. And there was just something that came out a couple of months ago from the East Coast that showed a butterfly that had been released, I believe it was in New Jersey, that got recovered a few days later in Georgia. So, that gives you some idea. Now, it's not going to be continuous, uninterrupted flight. They're going to pause to feed. They're going to pause if the weather's bad, that sort of thing. Mm-hmm. So it all kind of averages out to about 2 months to get there.
Yeah, it really is remarkable. It's such a small, what we would many people consider a very fragile organism, that it can travel so far.
So that is true, and yet, the monarch is the only North American butterfly that has a very well-documented migration with a starting area and a fairly defined endpoint. There are other butterflies that, as winter comes to this part of the world, kind of die back to far enough south that it's not bitterly cold, like the Gulf Coast or the desert Southwest. And then as spring works its way back up the continent, they'll begin kind of gradually breeding their way northward over the course of the summer. And depending on the species, it may get here in midsummer, it may get here right at the end of summer. It may not get here every year, but occasionally they'll be able to make it up this far. Well, one of the ones that is known, fairly common butterfly in parts of Illinois, is called the dwarf sulfur. And it's about that big. It's like less than an inch wingspan. And it's flying all the way up the Mississippi River from South Texas to get here every year. Mm-hmm. And that's much smaller and more fragile than a monarch is. So, it kind of depends on your perspective whether a monarch is actually small and fragile or not.
Right. Well, I think butterflies in general, I guess I'm trying to say, yeah.
Yeah, but it's still amazing that they're able to do that.
Yeah, for sure, yeah. Can you explain how mimicry and camouflage work in butterflies?
Sure. Mimicry and camouflage are both, in some senses, very different things because they're about the wing pattern, and in a sense, they're doing opposite things. They're having opposite effects. So camouflage is where a butterfly is often on the underside of the wings, going to be colored in a way that it's very similar to dead leaves or bark, or a lot of the bright yellow butterflies that you see. When they land and close their wings, they actually look quite green and blend in well with foliage. And so the idea is one of about blending in and not being seen by predators. One of the things that you will often find with butterflies that use mimicry as a defensive mechanism is that the upper side of the wings is often much more brightly colored. So question mark butterflies are a good example. On the underside of the wings, they look like dead leaves. They're sort of a mottled brown pattern. And on the upper side, they're bright orange. And the idea is the predator is going to overlook it, but if the predator starts getting a little too close, as the butterfly takes off, that flash of color may cause the predator to pause for just that split second that the butterfly needs to get away. And, it doesn't even have to work all of the time for it to be selected for by evolution. It just needs to work often enough that it's giving the butterfly a selective advantage to have that. Now, mimicry. Well, in order to talk about mimicry, you first have to talk about something called aposematism, which is warning coloration. And there, you've got a species that is in some sense, unpalatable to predators. And monarchs are probably the best known example, though there are certainly others in this part of the world. And so with the monarchs, the caterpillars are feeding on milkweed, which has a group of toxins called cardinalides in it, and they actually can sequester these toxins in their body tissues. And these toxins are retained through metamorphosis into the adult form. And so it's been shown by feeding monarchs to captive birds that haven't been exposed to them before. They will try to eat them, and then they will often get sick from eating them. And then after that happens, they will learn to avoid that kind of color pattern. So what you will find is that species that are aposematic will often have very bright, bold color patterns. Although for some reason black is also a pattern that is also found a lot in butterflies that are warning colored. And they will often also be less furtive than other species of butterflies. And because it's like, I don't care, you know you can't eat me, sort of thing. So mimicry is what happens when you've got more than one species having a very similar color and wing pattern. And there are basically two kinds of mimicry. One is Batesian mimicry, where you've got a model that's toxic and a mimic that's non-toxic but gets away with being non-toxic because it looks like the toxic organism. And for a long time, people thought that that's how the monarch and the viceroy worked. They both look a lot alike. They're both, orange with black stripes on them. And then somebody said, we've never actually tested to see that the viceroys, in fact, are non-toxic. And they did a similar experiment to what they did with the monarchs and found out, oh no, the viceroys are toxic too. The caterpillars there eat willows. There are a lot of alkaloids in willows. It's what aspirin comes from, so very bitter compounds. And so the kind of mimicry where you've got a toxic model and a tasty mimic is called Batesian mimicry. And the kind of mimicry where you've got a bunch of species converging on a common pattern is called Müllerian mimicry. And Müllerian mimicry probably works because efficiency. It's one pattern teaches the predators many more types of species in some cases to avoid.
What are some of the biggest threats to butterfly populations today?
For a long time, when people would ask me that question, I would say habitat destruction. Mm-hmm. Habitat destruction is still an important thing that's a threat to butterflies today. But we're starting to see declines in butterflies even in protected areas, even in areas where the habitat is being taken care of. And there are a lot of hypotheses at the moment, but I think we're still trying to learn the relative contributions to some of these things. So, certainly certain types of pesticide usage are going to, perhaps in different ways, be damaging to butterfly populations. Changing climate is almost certainly having an effect. And the reason we know that is because of things like the Illinois Butterfly Monitoring Network and other similar programs, where you look over a long period of time, decades in some cases, and you see changes in populations. And what we have tended to find is that there are species dropping out of the local fauna that seem to be at the southern edge of their range. And as the climate warms, the southern edge of their range keeps retreating northward. Mm-hmm. And you see these species really declining, and in some cases even dropping out of the area. And at the same time, a lot of these species that I was talking about earlier that migrate up from like the Gulf Coast each spring, a lot of them keep going further and further north. And in some cases, we're seeing ones that we have not really seen very often before. In other cases, there are species that it used to be unusual to see this far north that we're now seeing regularly. I think a good example of that is a little orange butterfly that becomes really, really abundant here in Chicago in late summer called the fiery skipper. And when I first came to Chicago in the early 1980s, that was a much more unusual butterfly to see here than it is today.
Looking back on your career, is there something that stands out as a pivotal moment in your research, or a discovery that stands out from?
A couple. One that was a pivotal moment, but was much earlier in life than I was doing anything that looked like research. When I was in college, I had become a little bit less active working with butterflies. And I found a field guide that I hadn't been aware of. I had always used the little golden guide to butterflies as my field guide growing up. And it's a wonderful field guide, and I still recommend it to people, especially people who are just starting out. But it's not complete. It doesn't have all the species. And so I got this new field guide, and there were species that I had never even heard of before in that, from the part of the country that I was in at the time, which was New England. And there was one butterfly mentioned that's called the bog copper, kind of a coppery color on top. And there were other species of coppers that I was familiar with, but I'd never heard of that one. And it said in the guide, it can be found in abandoned cranberry bogs. And I thought, oh, that's interesting. And so I asked my dad, I said, we go to Cape Cod a lot. Do you know of any abandoned cranberry bogs there? And he said, as a matter of fact, I know where there is one. And it's someplace near where we usually go. So the next summer we were on a trip, and he took me there. And I went into the bog, and I hit it at just the right moment, because this was a species of butterfly I'd never seen before, and there were dozens of them, maybe hundreds of them, all flying low over the cranberry bog. And for me, that sparked my interest in butterflies. It wasn't just going out with my net and seeing what I could find. It really introduced me to the notion of specifically looking for a butterfly's habitat. And that made the whole process a lot more interesting to me. It sparked my interest again. From a more research perspective, I think the thing that I am most happy with that we found is something that came out of the Illinois Butterfly Monitoring Network data. With the Illinois Butterfly Monitoring Network, you're looking at all of the butterflies that you find on the areas that you're monitoring, which include the monarch. There are other programs that are very monarch specific, but even though this one isn't monarch specific, we're still getting, a nice big monarch data set. And back in the 90s, you started hearing a lot about periodic snowstorms up in the mountains in Mexico that would cause mass mortality events. And, people wondered, what is this doing to the monarch population? And so I was thinking, well, we've got this data from the Illinois Butterfly Monitoring Network, so there's a way to begin saying, well, what's happening here long term? Do you start seeing a big drop-off in monarchs the summer after one of those big snowstorm events? And the answer is not necessarily. And what we started seeing, so you've got the data that we're collecting up here in Illinois each summer, and then there are data that are collected with a completely different protocol down in Mexico each winter. And you've got these two data sets, and you can see how they compare to each other. And what we found as we started looking at the two data sets is that what you see here in Illinois in the summer is a really good predictor statistically of what you're going to see in Mexico the following winter. However, the reverse of that is not true. What you see in Mexico in the winter is a very poor predictor of what you're going to see in Illinois the following summer, which leads to the hypothesis that for setting the annual population size, the reproductive success here in the summer is much more important than survival success through the winter. We did in 2015 publish a paper, myself and a couple of colleagues, that addressed that, and then there have been a bunch of follow-up studies that use way more sophisticated statistics, there I can say that, and have found additional details about how all of that works. And one of the things that seems to be the case is, for example, that generation in the Rio Grande Valley of Texas in the spring, how successful that is probably very important. Success in the southward migration is also probably important.
What have you learned from your research on butterflies that has informed something in your own life?
I think that what I have learned about my research in butterflies that's a good life lesson for myself is that you have to be patient, that things that look like they're happening in the short term may not actually be reflective of what's going on in the long term, and that it's just important to stay with it and not to give up too easily on what you're doing.
Some good life lessons, yeah. Has there been a work of fiction that you've read that has impacted your life and perhaps your research on butterflies?
Maybe not impacted my research so much as just one author that I really enjoy is Vladimir Nabokov. And he was actually, in addition to being a very famous author, was a lepidopterist. And he studied a number of kinds of butterflies, including the little blue ones that you see. And he developed a taxonomic hypothesis about some of these blue butterflies from Central and South America. And this was followed up in the 90s by a research team that found out that his work back in, I think it was the 40s that he did this work, was still sound, although they really expanded on a lot because they discovered a lot of new species that were not known when he did this work, and they still fit into the general pattern of taxonomy that he had. Some of his books, he loved wordplay and that sort of thing, and he will have these very sly butterfly references and butterfly puns and butterfly jokes in some of these books. And that makes them very fun to read. And he also wrote his memoir. And one chapter in his memoir is about collecting butterflies when he was a child, first in Russia. And he, describes what butterflies mean to him in a way that a writer of his caliber can describe in a way that somebody like myself can't. And I remember the first time I read that passage of his memoir being just blown away by it. And he uses the word ecstasy in the passage to describe how he feels when he is out in the field among butterflies and their host plants, just being out in nature at that time. And it's like, it was so on target.
Can you talk about the importance of citizen scientists?
Yeah, citizen science is such a cool and important thing at the moment. And there are two things that citizen science allows that more traditional approaches to scientific study have more difficulty with. And that is sending large amounts of people out to collect more data than can easily be collected by a typical researcher, even if you're counting the researcher, their graduate students, and undergraduates who are out doing it. And also, in academic settings, many, though certainly not all, research projects are limited by like a graduate student's period that they're working on their dissertation. And so citizen science, or as many people are now calling it community science, allows you to keep going and collecting data for a much longer period of time. There is one very famous, more academic study that does this. It was done by a gentleman named Art Shapiro, who was studying butterflies from a number of different sites at different elevations in California over a number of decades. But he is very much the exception on that type of thing. There just aren't many people replicating that. There's somebody who, after he retired, kind of took it over, but you're not seeing this happen in different parts of the country the way you're starting to see it with the community science programs.
What has been the most surprising thing you've learned about butterflies over the course of your research?
I think the thing that's been the most surprising to me is how much diversity there is. And by that, I don't just mean how many species there are, although that's certainly an element of it, but just how different they are in terms of their behavior, their range of habitats, their how they defend themselves against predators, what the range of host plants that the caterpillars feed on, and what the implications are of that for the ecology of the adult butterfly. And I also think how many things other than just host plants influence where butterflies can and can't live. So we were talking about community science. And the Illinois Butterfly Monitoring Network is a community science project that I have been affiliated with since its founding in 1987. And people often wonder when they hear about things like butterflies are faring poorly, some of the concerns people have about the monarch butterfly, what can I do? And participating in a community science programs is actually one of the really useful and important things that one can do. So the Illinois Butterfly Monitoring Network, what it involves is that when you sign up for the network, you get assigned to a place somewhere that you're going to do your monitoring. And on that place, we have set up a walking route. And you go back a minimum of 6 times each summer, and you walk the route. And while you're walking your route, you're envisioning yourself in a bubble that's 20 feet on each side of you, 20 feet in front of you, and the bubble moves when you do. And basically, while you're walking your route, you count and identify all of the butterflies that you see inside of the bubble. It takes about an hour to do each time, and a good site will see over time we'll pick up 25, 30 species of butterflies. So it's a manageable number of species that you have to get used to being able to identify. This has led to a really significant and large data set. We have currently over 21,000 surveys in the data set as of the end of the 2024 season. And increasingly over time, publications are coming into the scientific literature using this data set. And so it's telling us some really fascinating things. It's told us some things that we didn't know about the monarch before. It's been showing which species are doing well and which species are doing poorly, and in some cases implicating things like a changing climate. A paper came out about a year ago showing that site management actually does help butterflies. It's always been suspected that that would be the case, but it's another thing to actually show it. So there are very important things that the scientific community is learning from this data set that the citizen scientists are collecting.
Important work, yeah, for sure. Unless you had something else you'd like to add, I was going to wrap it there.
Okay.
Well, thank you very much, Doug, for your time. I appreciate it.
Thank you. It's been a pleasure chatting with you.
Great. Thank you.